Okay, so in continuation of our lab exercises today, day 3, it's finally dried, half an hour to dry. They must be completely dry before you fix them. So I did find a piece of dark paper, and I wanted to show you if I could get it up close enough without getting too much reflection. I think you can see it, they should be cloudy. There should be like a cloudy smear. I think on this bottom slide, this panel, you can see it's cloudy as well as this one. The top two are a little bit harder to see because of the angle of my camera, but they should have a little bit of a cloud to them in order to know that there are bacteria on there. Before we start, I wanted to show you my little apparatus that I rigged up here. I took 2 drinking straws and I just marked them with a Sharpie so that, and then I notched them out with scissors so that they would sit on the long side of this disposable container, the storage container, so that I can use it as a, maybe you can see the notch there, right? I marked it and then cut them out with scissors so that I would have a little staining rack. So they would lay relatively flat and I could put my slides on there. What's beneficial about this is then you can stain all of your slides at once. So I'm going to put my 2 slides that I have, and you don't need to do it over the sink this way as well. That's my motivation for doing it. So I'm going to move my straws as far apart as I can without dropping the trick is not dropping your slides in there once you have stain piled up at the bottom of the container. But I think you can see, I'll bring it a little closer, you can see how I have my slides situated on top there. Slide 1, slide 2. And these are the 2 that I've labeled SS for simple staining. Another thing I wanted to just go over with real quick with you, if you're trying to use your wax pencil and the tip is already worn down, you need to pull on the string and then you can unwind. I used these forceps. Then you can unwind the paper very slowly. I was gonna finish unwinding it in front of you guys. Very slowly unwind the paper, and then you get a new tip. And this is quite large, so this is too long. You can cut it with a kitchen knife. But I couldn't get, you can see there's perforations on here, but I couldn't get a single one, just a single perforation to unroll. So my tip is now super long. Okay, so I'm going to keep these forceps handy because I am going to use them to hold my slides over my ethanol burner, because you must fix your slides before you try to stain them. So a couple little tips about the staining. They must be dry, completely dry before you fix them. And how you'll know if they're dry is the whole area where you put bacteria and water will just look like a smudge. If there's any bright circle around the edge, that means it's still a little tacky or still has a little water in it. They must be completely dry before you fix them. You can stop after fixing and stain them tomorrow if you'd like, but they have to be fixed or they won't be good tomorrow. So just as they showed in the video, I'm going to hold them with my forceps and I'm going to wave each side. So first I'm going to take side B here and I'm going to literally just put it through the flame. And then I lost my flame. And I'm going to hold it over that flame. Wish I could adjust it. If we were in the lab, we would just zoom our slide through like this, maybe 3 times, kind of on an angle. And then when it's cool enough, I switch sides and I do the other side of the slide. These paraffin burners are so sensitive that I'm finding this to be a little difficult. You have to fix the slides. Let's just try holding it close. I think that's what they did in the video. So we'll do that for about, it's making a black spot, a few seconds here. Okay, let's see how that goes. If you don't fix, the bacteria will come off with the dye. So this may take a little trial and error. And since I burned my slides on the back, I'm going to have to try to clean that off before I proceed here. So I just took a paper towel and cleaned off that burned mess. And maybe this time I won't hold it quite so close to the flame. I think 5 or 10 seconds, about 5 seconds should be good. Be careful, I use plastic straws. If you use plastic straws, oh, you know what would really work really well? Probably chopsticks too. Anyway, if you put them down and the glass is too hot, it's gonna burn the plastic. So you're gonna have to be careful about that. All right, so this is the first time I've ever done this with a paraffin burner. So I'm probably fixing them more than I need to because I don't know how long I should really fix them. It's kind of trial and error. Now that they're fixed, I am going to turn off my ethanol burner or my paraffin burner because I'm going to be using Crystal Violet, which is dissolved in water, and safranin, which is also dissolved in water. But if we'd had methylene blue, I think that's usually dissolved in alcohol. So I'm just going to use the Crystal Violet first. I'm going to place that on, that slide 1 is going to be stained with Crystal Violet. Slide 2 is going to be stained with safranin. And I went ahead and put, you can see my Post-its here. I put the timing for simple stain on a Post-it in front of me and the time for a gram stain and the different reagents in front of me. So I can just look up and not have to fumble through my handout or flip through my computer to find exactly how long each thing needs to stay on. So I give this a little shake, and then the simple stain says Crystal Violet for 1 minute. So I'm going to just turn on my dropper bottle and I'm going to place a nice big drop of Crystal Violet so that it covers my smears. And I'm actually going to put the safranin on the same time the other slide, slide number 2 here. I'm going to use my watch for a 1 minute time. Now it seemed a little light, so I just put a little extra drop on that. These times are always minimum times. So if it's on there for a few seconds longer, 30 seconds longer, that's perfectly okay. You don't have to worry. Please don't use a hair dryer to dry your slides. It'll be too hot too fast and it will cause the cells to burst, and then they won't be able to be seen very well under the microscope. So I've got another 5 seconds here. That should be good. That's a little over a minute. I have a dropper already placed in my distilled water, and I'm just going to squeeze the bulb, submerge the tip till I fill this all the way up. It's a little over 1 ml. And I'm going to just rinse these slides one at a time. I'm going to tip them up like this so that, and I'm going to tip them so that the dye runs right off of the slide and into my waste container. Now, water is polar covalent and glass is positively charged. So you might be able to see, might not, that I've got a lot of dye built up on the back of my slide. So I'm giving that a little rinse as well. You want to not touch the smears with the tip of your pipette because you don't want to contaminate the pipette, because I have to put that back in the distilled water for the other one. I'll let that one just rest there for a minute. Maybe I can turn this way and you guys can see a little better on the second one. So again, I'm going to tip see how I have my 2 smears? I'm going to tip them away from me, away from my body. And I'm just high above each smear. I'm putting a few drops of water, tapping. I'm going to grab some more water, and I want to be sure to get the stain that's accumulating on the back of the slide. I want to be sure that gets removed as well. All right. Place my pipette into the distilled water container. And now I need to blot these dry. To do that, I'm just going to take half of a piece of paper towel and put it down on my lab bench here. And I'm going to place my slides on it like that. And then you want to put the paper towel over the top and see how I'm just very gently patting them. Nice little pats. Oh, nice little bacteria. Don't do this. You'll take the bacteria straight off the slide. and don't press down too hard. I'm on tile, so if I press down really hard, I'd probably break my slides. And some of the dye might come off on the paper towel. That's okay, but just I wouldn't keep pressing that. I would say you're finished. And I'm going to try to hold these up so you guys can see. Slide number 1 was stained with, which is actually on the bottom, was stained with Crystal Violet. And slide number 2 was stained with Saffron. And that one's a little bit hard to see, but they are pink. They are slightly pink. The Crystal Violet is a much darker slide. So I'm going to set these aside because myself, I'm going to look at all my slides under my little microscope at the same time. So I'm going to put these to a slide. I might write on this paper towel, simple stain, exercise 3. And I'm going to set those somewhere out of the way. And then I'm going to keep using my waste container here for my gram stain. I'm going to make the gram stain a second recording. I think I'm going to pause the recording for a couple of seconds and then we'll go on with the gram stain. All right, I went and retrieved my other set of slides that I had set up on the little different container here. And these are the slides for the gram stain. So I'm going to remove this out of the way. And again, these slides are also going to need to be fixed. So I'm going to light my paraffin candle again. And I'm going to hold the slides with my forceps so that my gloved fingers don't get too close to that open flame. And I'm going to hold them over the flame about that high for I'm going to give them each a good 5 seconds. Remember, if you hold it too close, they're going to burn. And if you hold it too far away, they won't actually be fixed. So it's kind of a trial and error. In the laboratory, we would have what we call a slide warmer, which is basically like a hot plate. We set it on a low setting, and the slides can be warmed gently to get the bacteria to stick to the glass. The purpose of the fixing is, the air drying is to try to get them to stick to the glass, and then when we fix them, it's actually fixing them to the slide so the bacteria stick to the glass more tightly than they stick to the dye, as well as so that you kill any bacteria that are still alive through the drying process. This is my tooth and gum scraping, so I'm giving that one a little bit extra. Looks pretty gross from there. What have we got here? This slide looks like it's cooked a little bit. Oh, that's the serratia, so maybe those weren't as dry as I thought they were. Let's give that guy a few more seconds. I have no idea how these are going to turn out, so we'll have to take a look at them under the microscope. And then if they're terrible, I'm going to wash the slides off and do them again before I photograph the pictures and post them. I'm going to do the gram stain in a similar manner as I did the simple stain, in that I'm going to do all the steps to all 3 slides at the same time. It may seem a little stressful for you at first, and I understand that, but the advantage is that you're finished with each step one at a time and you don't have to repeat them. And this way also you can actually, it's a little more time efficient because you can do all of them at the same time. So I'm going to blow this paraffin candle out because we're going to be using ethanol, and it's 95% ethanol, and we're going to be using that for our decolorizer, and we want to be sure that there's no open flame anywhere near that. So since it took so long for my slides to dry, I recapped my reagents, so I have to very quickly here open up the ethanol again and the grams iodine. All right, so my gram stain directions are here abbreviated. I just put CV, 60 seconds rinse, iodine, 60 seconds rinse, ethanol, drip, rinse with distilled water, and saffron, 60 seconds rinse. Again, remember these times are minimum times, so it's okay if you actually end up putting leaving the stains on for a little bit longer. And I'm just going to go ahead and put drops of Crystal Violet on all 5 of my smears here. So I'm just moving so I can see where the smear itself is. And I'm just going to put some drops on. And I know it says 5 drops, but my smears aren't that large, so I'm not going to waste the dye and put 5 drops on. Start my timer for 1 minute. And as you are already well aware, I'm not good at skipping the boring parts in the YouTube videos, so you're just going to have to watch for a whole minute while I wait for my stains to have enough time on the cultures. A minute seems like a really long time all of a sudden. I could stand up my notebook back here. Make it a little more interesting than looking at a blank wall. Okay, that's 1 minute exactly. So it says after 1 minute, I'm to rinse that with distilled water. So I have my cup of distilled water with my bulb, my pipette in it. And again, notice I'm tipping them away from me. And I'm very quickly here, I'm just going to move my hand and try to rinse all of them once, right at 60 seconds. And then I'm going to rinse them again. Remember, you want to hold this pipette way high above the slides because you do not want the tip of the pipette to touch the slides. If you transfer stain into the water, potentially you could have transferred bacteria into that water as well. So you're going to have to get yourself a second cup of water. If you're fearful that that might happen, it might be wise to have a second cup of water, of distilled water, ready to go. If I had a squirt bottle, this would go a lot faster, which is what we usually use in the lab. Okay, all Crystal Violet off. Now I put the iodine on and let that sit for 60 seconds. We do not dry the slides in between these steps. So I am going to use a lot more iodine than I did of the Crystal Violet because now my slide is wet and the iodine is a little bit diluted because there's still wet water from my rinse on there. So I'm putting a little bit more iodine on. That needs to sit for 60 seconds as well. So ready to go. I would be sure to be wearing your lab apron today and old clothes that you don't care about in case you get dye on you. I'm of course wearing my lab coat, but it's up to you. You can do whatever you want. I would definitely wear the lab apron. The safety glasses are included. I definitely put gloves on today. The safety goggles, because we use such small quantities, it's rare that anyone gets any of these chemicals in their eyes, but if you feel more comfortable wearing them, by all means wear them. Just a few more seconds. This next step, the ethanol rinse, this is the hardest step. This is the step that most students, it ruins their gram stain. So I'm going to encourage you to be very careful, and I'll go through it as we are actually doing it. So first thing we want to do is we want to let the the iodine sort of run off of our slides, and I'm going to tap them a little bit. I'm going to do that to each slide so that they all have the iodine on for about the same amount of time. And now I'm going to take this ethanol, and this is, like I said, a really important step. We're just going to put a drop on, and I'm going to tap it off, and a drop, and I'm going to tap it off. A couple drops, tap it off. And then I'm actually knocked my bottle over. It's a good thing they gave me a second bottle of alcohol. Then I'm going to rinse this and rinse the excess ethanol off immediately from slide number 1 here. And the reason I'm doing that is because I don't want the ethanol to sit on there for too long. And how you know that it will be completely rinsed is at first when you start to rinse, it's resisting the water, but after a couple squirts, you get a better adhesion. So I'm going to dribble, 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 tap, tap, dribble, 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 dribble. Sort of rock my slide a little bit, tap, tap. And you notice they don't give a time for this one because it's really like 5 to 10 seconds. And then I'm rinsing with water. Try to turn a little bit so you guys can see what I'm doing a little better rather than being so much on the side. Okay, that looks good. Now I'm going to take a little more ethanol and I'm going to drip, drip, tap, tap, drip, drip, roll it around my slide, tap, tap, drip, drip, tap, tap. I'm going to call that finished. And I am going to cap this since I made such a mess with the ethanol. And I think I'm on slide number 3 here. So you can see it kind of, I don't know if you guys can tell, but it kind of resists the water at first. And then after about on the 3rd rinse, you can actually see that it's rinsing. Okay, I'm just going to give these guys an extra squirt just for good measure here. Couple taps. And I am going to clean up that alcohol that I spilled. Because when I light my paraffin burner tomorrow, I don't want everything to catch on fire. That would be bad. So yeah, it looks like I lost about half the bottle, but remember we have more. We actually received 2 bottles of denatured ethanol at 95%. And worst case scenario, if I dumped both of them out by accident, you can get alcohol at a drugstore, at a CVS or a Target, 95% instead of 70. So I would be able to replenish that. All right, now I'm gonna give my saffron in a little shake. This isn't a dropper bottle, so I'm just gonna put a drop or 2. Again, now that there's water and alcohol in here, I want to be sure that I really cover the smear with the saffron. And so I am going to put a couple drops on each smear rather than just a single drop like I did with the Crystal Violet. Put the cap on. That's the best way not to spill them. All right, so I am going to take the pipette out of my Graham's iodine whilst I'm watching my timer here. That Graham's iodine will stain as well, so be careful when you move that pipette around. And that's why I put mine on a little bottle, a little piece of paper towel, so that it wouldn't get on my countertop. Almost done. So this is almost the last step, and then we can let these dry, and we can look at them tomorrow. Maybe about another 5 seconds. Okay, that should be pretty good. This gets easier with practice. So, like I said, I believe you have enough supplies that you could probably do at least 2 sets of Graham staining, which is why we made those other slides ahead of time. Mine are still laying here drying. I did not make a spare tooth scraping, but I did make a spare slide 1 and slide 2. And I'm just going to rinse these until there isn't any more pink coming off of the glass. Oops, got that on my dark paper. And now it would be probably easier for you to see the bacteria if I showed them to you on a piece of plain white printer paper, which I do not have at hand. But I have a paper towel, so let's see how these guys look. I lost most of my markings on the slides when I put the alcohol on them, because remember, the alcohol will dissolve the Sharpie. So I can still see that, you guys can't see that, but I can still see that I've got slide 1, slide 2, slide 3 here, which is good. Slide 3, remember, only has 1 smear on it. So that's going to be pretty easy to remember. I've lost what bacteria were on here in what order, but I wrote it down, which is important. So I'll be able to, I just see like little scratches there. So I'll be able to look it up. I'm going to pat these dry gently. And I don't know. how we did here. That slide looks pretty grim. So I'm gonna, remember you can wipe the back sides of the slides and you want the back sides to be clean. And really dry. And then these top sides, best to do is just to pat them gently like that. And I'm gonna let mine sit now for probably an hour or so. I'm gonna go eat dinner and then I'm gonna come back and take a look at them with my microscope. Oh, as for cleanup today, I'm gonna just keep my stain set out. I'm probably going to switch out my pipettes because I use them. And we have several, several came in your kit. So I'm just going to use a new pipette if I have to gram stain again tomorrow. Same with my distilled water pipette. I'm going to dispose of that. I'm going to dispose of my distilled water because it won't keep. It looks like I'm almost ready. I'm going to need a new bleach solution. I'll make that up tomorrow. This should be plenty to clean off my workspace today. And I am going to dispose of all of our things from day 1 and day 2. That we're supposed to sit in bleach for 24 hours. The 2 vials and some of these pipettes have been sitting in here for 2 days now. So I'm going to take these downstairs to my kitchen sink. I'm going to have the sink completely empty. And I'm going to run this bleach with the dead bacteria down the drain with lots of water. And I'm probably going to flush each of the pipettes out a few times before I dump the bleach cup and then with water before I throw them in my regular trash. I'm also going to dispose today of all of my inoculating loops. This is almost all the ones that came in that first bag and my toothpick down there. And these, basically, I'm just going to swirl them a little bit, and then I am just going to pop them into my regular trash, put the alcohol down my kitchen sink with lots of running water. It looks to me, even as we were talking, as I was spending the hour or so doing this, some of the bacteria are, Serratia is pink, and it's starting to turn pink. You can't see it, but so these plates will be ready to read tomorrow. So I'm gonna leave those set aside along with my slants and broths. I do think that my bacteria could probably benefit from being transferred into fresh culture, especially my Serratia and E. coli. Can you see how much more cloudy those 2 are than the other 2? So I am probably going to take a loop full of each of these and put it into a fresh nutrient broth tube. The container came with 10 broth tubes, and you can see I've only used 4 or 6 of them. So I do have a few broth tubes left. So I am going to do a subculture of these 2, of E. coli and Serratia. These 2, the Micrococcus and the Staph, still look pretty good. Okay, see you tomorrow.